Let's go through everything you need to know for GCSE Biology Paper 1 tomorrow, part 1. And if you don't already know me, my name is Chris and I'd really appreciate if you repost this, send this to your friend and drop a follow. Now, eukaryotic cells are cells like your plant and animal cells. They have a nucleus, a cell wall, if they're a plant cell membrane for all of them, cytoplasm, and they have membrane-bound organelles. So like the nucleus, for example, has a membrane around it. Whereas prokaryotic cells, things like bacterial cells, have a loop or small rings of DNA. Plasmids are circular pieces of DNA. They have a cell wall, cell membrane, cytoplasm, and no membrane-bound organelles. So the DNA is not enclosed within the nucleus, it's floating around in the cytoplasm as plasmids and chromosomal DNA. Also remember that plant cells might have a cell wall made up of cellulose, but prokaryotic cells usually have a cell wall made up of a protein called peptidoglycan. Prokaryotic cells are much smaller than eukaryotic cells. Eukaryotic cells usually range from about 10 to 100 micrometers in size, whereas prokaryotic cells are usually 1 micrometer or less. Moving on to specialized cells, and this one is a nerve cell, and you can see that it's very elongated, the axon's super long, so that it can reach and send information from the brain all the way to the different parts of your body. Now, the function of nerve cells is to conduct electrical impulses around the body, and the way that they are adapted to do this is this long axon that we just mentioned. And you can see that the axon has these layers of myelin sheath, which is a fatty insulative layer, which allows for the electrical impulses to move faster across the body. They also have these extensions of the cytoplasm called dendrites at the end here, which allows them to communicate with other nerve cells, with other glands, to help transmit that information to all the different parts of the body. These specialized cells are muscle cells, and you can see over here that they are filled, absolutely filled with mitochondria to allow for respiration to take place and therefore for a large release of energy to help keep those muscles contracting. Muscle cells are also made up of these protein filaments which slide over each other, contracting the muscles, which is another adaptation for them. This specialized cell is a sperm cell, and at the tip of the head we can see that there is an acrosome. The acrosome contains digestive enzymes which help to break down, digest the outer layer of the egg cell, which is necessary for fertilization, because that's where this nucleus of this sperm cell provides the haploid set of DNA to the haploid set in the egg cell, allowing for the fusion of those and therefore fertilization to take place. The other adaptations of the sperm cell allow it to actually get to the egg cell. So the mid piece over here contains many mitochondria. Those mitochondria release energy through respiration to help this tail, this flagellum, to move and propel the sperm cell towards the egg cell. This over here is a root hair cell found in plants, and its role is to maximize the absorption of nutrients and water and mineral ions from the soil. Once again, we can see that there are mitochondria present. Wherever there's mitochondria, there's a release of energy, and this energy is necessary for active transport, which we'll talk about later. But active transport requires energy to move things from an area of low concentration to an area of high concentration. We can also see that root hair cells have these long extensions which allow them to increase their surface area and therefore increase the surface area over which diffusion or active transport can occur, therefore increasing the uptake of mineral ions and water. And we can also see that they have very thin walls over here, which decreases the diffusion distance, making diffusion, osmosis, active transport all easier. This over here is a xylem, and xylem transport water and mineral ions up a plant, only up. And xylem are made up of dead cells, meaning they don't contain the organelles that you'd expect in a living cell, and they don't contain the cytoplasm either. This maximizes the amount of space available for water and mineral ions to be transported up the plant through the xylem, but it means that they are weaker, which means that they require linen or lignin to help structurally support this vessel and avoid it from collapsing. Because these are dead cells, there's no mitochondria, it means that a passive process has to take these up the plant, and that passive process is osmosis. And that osmosis upwards movement is created by the water evaporating from the leaves at the top of the plant, drawing up the water and mineral ions up this xylem vessel. The other vessel we need to know about for plants is the phloem vessel, and this one is made up of living cells which also have companion cells next to them filled with lots of mitochondria because they require energy. And phloem vessels are involved in translocation, which is movement of dissolved sugars and amino acids up and down the plant wherever they are required. And whereas a xylem vessel was a long continuous tube, this phloem over here has sieve plates in it. Now, sieve plates have holes in them, which allows for easier transport of those dissolved sugars and amino acids. Moving on to microscopy, and we should already all be familiar with this diagram of a light microscope. And if we wanted to view a sample of a specimen under this light microscope, we would firstly prepare this slide, which would mean placing the sample on, perhaps dyeing it if that's necessary, placing a cover slip on, and then placing it on this stage. Once the sample is clipped into place and it's secure, we can then turn on the microscope on the lowest objective lens. So we turn this on onto the lowest magnification and we'd look through the eyepiece. We would see a blurry image, which is where we'd use the coarse adjustment knob, which is situated over here. This would clear up that field of vision, and then we'd use a fine adjustment knob to make final necessary adjustments to the focus. If we then wanted to see the image at a greater magnification, we would restart the process by changing this objective lens to a higher power lens, and then using the coarse adjustment knob and the fine adjustment knob all over again. And when you have to deal with calculations in microscopy, remember this triangle I am. And it's really simple. All you have to do is cover up whatever you are looking for. So let's say you're trying to calculate magnification, you'd cover up the M, and this triangle shows me I then have to do image size divided by the actual size. Something that's very common here is that examiners might give you two measurements, one in micrometers and one in millimeters. Before you do any calculations, it's best that you convert them all to the same unit before sticking them into this equation and getting the answer wrong. And remember, millimeters to micrometers times 1,000, micrometers to millimeters divide by 1,000. So 1 millimeter would be 1,000 micrometers, and 1 micrometer would be 0.001 millimeters. And I'll take a breather before releasing part 2. Make sure you drop down any requests for specific topics in the comments and tag a friend so that they can watch this before their exam too. And drop a follow.